Okay, thank you very much for the introduction of Alex. And hello, everyone. I'm Frank. I'm really excited today to talk about our work on the dual-purpose target discovery, as well as the aging research with our precious GPT models. Yes, this is the paper that we published last year in Trends in Pharmacological Science that we talk about how AI can help in the drug discovery, including, for example, the target discovery, indication expansion, and drug repurposing. Okay, and the paper was also chosen by as the editor's choice last year. So since 2020, we can see that more and more AI-identified targets has been being validated with wet lab. And one of the targets has been even moved to the human clinical phase 2. That is our TNIC targets. So it's a very exciting, okay. And then this is our Insilico approach, how we do the target discovery. So basically, we use multiomics data and text data. We use our 23 AI and bioinformatics models to calculate the score for the interactions of the disease, genes, and the biological process in real time. So that, let's say, we can start from the disease of interest, and then the system will help us to provide a list of the target candidates for the best target for the disease. Or we can start from the target of interest, and then the system will provide a list of the best indications for that particular target. So that is the way how we do the target discovery, indication expansion, and drug repurposing. So early this year, we published a paper that is our lead program, the TNIC for IPF on the Nature Biotech Journal. So as far as I know, it is the most successful and the most comprehensive story example to show how AI can really help drug discovery. So you can see that since 2019, we use our AI platform Pandomics to identify TNIC as the best target for IPF. And since IPF, the fibrosis is kind of an age-associated diseases. So that's why we also do the hallmark of aging assessment. It turns out TNIC has been implicated in several hallmarks of aging. For example, the altered intracellular communications and cellular senescence. So once we confirmed the target, and then we use our generative AI, the chemistry 42, to design truly novel molecules to tackle this target. And then we went through a series of the in vitro, in vivo preclinical experiments, and then we moved to the clinical within 30 months. So this is another industry record that it moved very fast. And now we already finished the phase 1, and we have moved to phase 2 in both China and US. So since we identified TNIC in 2019 as the best target for IPF, and then we truly believe finding the therapeutic targets implicated in aging is very helpful. So that's why we try to use our Pandomics to very cost-effectively to find the targets indicated in multiple aging disease, because we believe that aging is the driving force for most of the diseases. And then we can also see TNIC is implicated in several hallmarks of aging here. After we identify the targets for multi-age-associated disease, we further classify them into different hallmarks of aging. And then last year, building on this momentum, we focus on whether we can find a dual-purpose target for the treatment of cancer and aging. So that's a very interesting. So we collaborate with Evandro from University of Oslo to focus on this study. So basically, we try to identify the targets that is commonly implicated in 11 solid tumors. Those are the common cancer targets. And then we also use the GTX RNA-seq data to find the targets associated with aging. And then we classify those targets into several groups. So you can see that the green targets are those with evidence in extending lifespan. So here, our definition of the dual-purpose target is that with the same direction of interactions that we can achieve the goal for the treatment of cancer at aging at the same time. So probably you know the best example is the mTOR, the rapamycin example. So however, you can also see that there's over 100 cancer targets that do not have the known effects on lifespan. So next, we are going to propose some dual purpose targets. We propose 23 of them, and the most interesting one is the KTM1A. So from this figure, from panel A, you can see that KTM1A is upregulated in 10 out of 11 solid tumors. At the same time, KTM1A is negatively associated with age. I mean, its expressions. So you can see that KTM1A is downregulated. So I'm not surprised because, you know, mTOR also have the similar situation. So that's why we want to see whether the knockdown of KTM1A can really help to increase the lifespan. So that's, we did the RNA knockdown experiment in the C. elegans, and the result is very impressive. So it is a very good example to show that how AI can facilitate the identification of the dual-purpose targets. So yeah, this is the story I want to share today. And now I will pass to my colleague Fedor. He will introduce very interesting GPT models. Thank you.
Thank you. Thank you, Frank. Hello, my name is Fedor, and I will go a bit more into detail about PreciousGPT. Previously, a little bit spoke to us by Alex Sharonkov during his talk, although I guess that time PreciousGPT was not the highlight of his presentation, but now we'll talk more about Precious3 and how it can further our advancements into generative medicine, drug discovery, and probably also help you in aging research and many other settings. So, first of all, PreciousGPT is a live model, by which we mean that it is intended and we aim to use it to capture the life in all its complexity with all the different modalities, including omics, including cross-tissue compatibility, and also we want this model and the whole family of models to be applicable to basically any tissue or living systems, such as cell lines, tissues, or organisms. As previously mentioned by Alex Sharonkov, the concept was first conceptualized and inspired by Gato, which is a very powerful architecture that is capable of interpreting 1,000 different data types and also providing output for 1,000 different contexts. And PreciousGPT is our last iteration in a whole family of models, which first was published about roughly a year ago with Precious1GPT, which is a transformer-based aging clock. Precious2GPT has just been published in Nature Aging, and Precious3GPT has recently been released on bioRxiv, and you can access and read more about the validations that we did with this model by following this QR code. And for Precious3, we actually have a range of activities planned to follow this conference. We are looking for community engagement. We are open to collaboration and helping you design digital experiments using Precious models and introducing generative AI and synthetic data into molecular biology and anti-aging research. Now, let's talk a little bit about what Precious3 technically and actually is. To train Precious3GPT, we used a large corpus that we constructed ourselves of publicly available omics research datasets. We used an aggregation of data sources such as Geo. We used also some of our own products to build this corpus, such as Panda Omics. Lynx was a major part of our training training set, since it contains so much useful information about how expression and other chemical perturbations and genetic perturbations affect expression levels in different cell lines. And we are trying to integrate Precious3 into a wider range of our activities, such as constructing a multi-agent system, which is ultimately intended to completely replicate the standard workflows typically encountered in biological research. We envision Precious models able of, at least partly, reproducing the capabilities of scientists and researchers who can put together scientific hypotheses and figure out the experiments to validate them and further use the generated output to ask further questions. And some of the specific tasks that Precious3 GPT is capable of carrying out are listed here. You can use it to discover natural compounds that would protect you from aging. You can do general target identification, drug discovery, and basically anything that you are currently doing with real omics data could be replicated with Precious3 GPT. Internally and mechanistically, Precious3 GPT is trained on not just omics data, which is ultimately tabular data type, but also on text data and knowledge graph data, which means that it is multimodal, not just in the sense that it is multi-omics, but it can use completely different data types to build abstract representations of biological entities. And the actual input for Precious3 GPT is formatted as text. This model is not conversational, although its input is a list of various conditions that you can define. For example, you can define the tissue, you can define the species. Currently, it supports three types of species: mouse, human, and monkey. You can include into the list of your conditions diseases, various compounds, the dose of the compound administered, the time of exposure, biological sex of your subjects. You can also include various cell lines, hundreds of them. And finally, you need to specify the instruction which will tell the model what is the research setting you have placed it into. That is, we have different instructions to replicate, for example, in this case, the case control omics study in which you are trying to assess the effect a disease has on differentially expressed genes. But there are other instructions. You can directly skip this stage of omics data generation and instruct the model to provide you with the compounds that can exacerbate or alleviate a condition. What happens with the prompt is that you leave the fields of interest, you leave them empty, and the model attempts to complete the input prompt to the best of its ability. Currently, the drug applied to the system is missing, and the output will have the same prompt, but it will also have candidate drugs that can replicate what else is said in this prompt. For example, drugs that can replicate this omics signature. Now I'll show you just a quick demonstration of how actually we can use these different instructions and Precious3 GPT as a model to find novel anti-aging compounds. So first we define the setting to be reproduced with Precious3 GPT as case control study in which the control group are young adults and the case group are older adults. You ask Precious3 GPT to generate differentially expressed genes, but not necessarily expressed genes, since you can use any type of 3 omics, proteomics, also methylomics, so you can ask for hypo or hyper methylated genes instead of differentially expressed genes. Once Precious3 GPT has finished generating this aging signature, You can actually reverse the signature and ask the model which compounds could replicate these reversed signature, thus negating and counteracting the flow of time in a biological system. And these compounds were tested in our robotics lab in an in vitro setting with IMR90 cells. We used rapamycin and navitoclax as controls for synomorphic and senolytic activity. And in the end, we obtained we shortlisted 22 compounds based on PreciousGPT's output, and out of them, 8 showed synomorphic or senolytic activity. And we are planning to look deeper into these compounds and explore the range of concentrations in which we tested these compounds to maybe find the optimal therapeutic window for these molecules. And the most exciting part about PreciousGPT is that it is an open access model which you can access on our Discord server. It is a demo version with limited capabilities, but you still can touch it and play around with it. You can also follow us on Twitter and X to get the latest versions and updates on our activities with Precious 3. But most importantly, we placed this model on Hugging Face. We also released a test dataset with some compounds, so you can test PreciousGPT and maybe come up with your own experiments, and the implementation of your ideas is as painless as possible. We released demonstration notebooks that you can just download, run, and easily modify to express your research ideas. And to demonstrate that PreciousGPT is easy to integrate with other AI tools and multi-agent systems. Here we have Anton Kolaga, our good friend, who has managed, together with his team, to implement PreciousGPT as part of their Longevity Genie chatbot. And now you can talk to PreciousGPT and apply other tools to its output that are present on Longevity Genie. And this concludes my part of the presentation. You can, hopefully this QR code works, so you can download the whole presentation and explore it on your own time. Thank you.
Fedor, you're not going to propose on stage today, right?
Not to Frank.
That would be a lot of fun, because actually most people don't remember what I was talking about. So this model is available open access. Again, I didn't even know that Anton Kolaga managed to do this. So maybe you can stand up. So this is really cool. You know, they did it during this conference, so it's actually so easy to, yeah. Yeah, so they managed to do it in a day. So our idea is to ensure that it's a community effort. You can utilize this model as much as possible. We're not making money on that. It's not a plan. We actually want to try to integrate it in the community. We're going to be building bigger life models. And at some point in time, we're going to publish a community paper. And hopefully it's going to be high enough journal to go after. Currently, it's on bioRxiv. It's with Vadim Gladyshev, one of the, when you ask ChatGPT, name top 3 aging researchers in the world, it always comes up with Vadim Gladyshev within top 3. So that's why during GRC Systems Aging Conference, when we were first kind of conceiving this idea, we decided to collaborate with Vadim's team, and his group is also doing experimental validation and are on a paper, so it's actually kind of maybe 50% his model, just as 50% ours. But when we publish, we really want to publish as a community. So now we'd like to open the floor for maybe, Morten, how many questions do we have? One, two? But these guys are on site, and they're right there, so you can always talk to them. They're like super accessible. Let's do one question. All right. Is it like smart question? Let's go.
Thank you. Tremendous respect to this work. I'm wondering if you could expand upon your benchmarking and data cleaning protocols for the data that goes into this model.
Sure. Let me roll back a bit. Can I roll back? I can.
Ooh, I see you have like all kinds of interesting websites open. I'm making a joke.
Okay, yeah, so the important thing to remember about PreciousGPT is that it does not accept raw data, no raw expression files or CPG CPG methylation values. It already requests partly processed data, which represents differential something, differential omics. So basically its input are 2 gene lists with some meta variables. These are abundantly available in the data sources that we used. Particularly, as I mentioned, links is the major part from which we draw information, omics information about how a compound affects living systems. There they have what is called level 5 data, which basically is already processed down to the point of being 2 up and down regulated lists. And we applied similar formatting for all other data sources. And in further iterations of Precious, currently we do not do that, but we definitely need to include more robust annotations. So currently you can probably see that we do not use information about the platform or the producer or the lab. These are not parts of the prompt, but I agree that these could greatly help us improve the cross-compatibility with legacy systems and, I don't know, systems to come.
Thank you, Fedor. So we will be able to talk to him right outside the conference.
Yeah,
and
I'm always
just
at the booth.
if the question was about benchmarking in terms of the model performance, oh, no, okay, so he answered it properly. But for the model performance, we actually did multiple experiments with state-of-art models, and in many state-of-art models it outperforms. We didn't put all of them here at this presentation because at some point in time we want to publish some new really cool stuff in a big paper. So some of the things are not yet disclosed.
All right, thank you so much. That was really fantastic.